So our next thing that we're going to be talking about is something that we've already kind of talked about, but now we're just talking about it more in depth and with more detail than we did before, because in order to build off of it, we got to learn a little bit more about it. So these notes are going over DNA structure and function. So we have talked a little bit about DNA, but like I said, we're just going to talk about it even more now. So you should have your notes in front of you, ready to fill them out like always. So the structure of DNA, it is double-stranded. And it is helical. That's what that word is. And that just means it forms a helix. And it's a double-stranded helical nucleic acid. That's very important. That's the molecule of life that it is. So remember, we have those 4 molecules of life: carbohydrates, lipids, nucleic acids, and proteins. So this is one of them. This is an example of one of them is DNA, and DNA is super important to us. Another detail about them is that DNA molecules are the polymer. So polymer with poly meaning many. So when we say that it's the polymer, that just means that there are many things making up the DNA molecule. There are many little pieces that go to build the molecule. So DNA molecules are made of the monomers, because mono means one. So the things that the polymer is built of are the monomers, known as nucleotides. And these nucleotides are made of 3 things, and they form a nucleotide. So these 3 things are, the first one is nitrogenous bases. Nitrogenous, meaning that they consist of nitrogen, okay? So you can say nitrogenous bases, or a lot of times you'll just hear me say bases. And these 4 bases for DNA are, the A is adenine, the C is cytosine, the T is thymine, and the G is guanine. So those are the 4 bases. Those will repeat in patterns, in specific patterns, not even in patterns, sometimes just they'll repeat in sequences rather. And those bases help make up the DNA molecule. The second thing making it up is the 5-carbon sugar, known as deoxyribose. So we've talked about other types of sugars, including glucose, we talked a lot about. So deoxyribose is just another type of sugar that goes into making up DNA, because if you remember, DNA stands for deoxyribonucleic acid. So in deoxyribonucleic acid's name, it tells you what type of sugar and what molecule of life it is. So that's pretty handy for us. So the nucleotide is made of the base, either A, C, T, or G, the deoxyribose sugar, and finally a phosphate group. And that's just what we'll call it. We won't talk a lot about the structure of it. It's just a phosphate group that connects to the sugar. And it's obviously made of phosphate. So these nitrogenous bases, since DNA is double-stranded, we know, we're going to draw a quick picture of DNA over here. So we know it's helical, but say we took that helix like a spring and just untwisted it, what would it look like? Well, if it's untwisted, it's just going to look basically like a little ladder, okay? That's That's the simplest way we can depict it to see the three things that it's made of. So basically this structure is just if we take the DNA double helix that you see, and it's basically like a twisted ladder. Okay, if ladders could twist, that's what it would look like. If we untwisted DNA, this is how it would basically look like. And so if we're drawing it, and I want you to draw this in your notes, or you can get out the structure of genetic information worksheet that you colored the other day, and you can be marking on that to kind of illustrate what different parts of the DNA we have. So, but at the very simplest form, this is how we're going to draw DNA, because it's very difficult to draw it in the twisted form and still get all of the information from it. So on our little DNA, whether you're drawing it in your notes or drawing it on the worksheet that you have, the structure of DNA that you have on your paper. So these right here, these are going to be your bases. So there's one base connected to, and then this backbone right here, we'll call it the backbone. That's where the deoxyribose and the phosphate group are. So the bases are connected to the backbone, which is the phosphate and deoxyribose. And so this side is one strand. That is one strand. And then the other side is the same thing. Just the complement, which we'll talk about. But this is also a base. And then this entire thing is the deoxyribose and the phosphate group. Okay, and these bases right here that we have are connected to each other, like how the strands stay together. They're connected or paired by weak hydrogen bonds. They're paired by weak hydrogen bonds, and the part about them being weak is very important, and we'll kind of hint back to that in a little bit. But they're just paired together by a little bond over here that connects them together so that the two strands are bonded chemically instead of just floating around separately as one strand each. Okay? These strands run anti-parallel to one another. Meaning the 5' end is parallel with the 3' end. So this is all kind of confusing if we're just basing it off of this picture. So I do want you to go ahead and get out your structure of genetic information worksheet that you should have in your possession. Go ahead and get it out because we are going to mark on it through the video. So you should have this paper in front of you and ready to mark on it as I am saying. So let's say, let's go ahead and assign these colors. Yours should be colored better than mine because granted I am doing it on an iPad. So let's keep that in mind. So there's our color of our adenine. Let's see, our thymine can be this color. And let's see, our cytosine can be yellow. And then our guanine can be a green color. Okay, so we should have those colored according to the diagram. So I'm going to go do that real fast. Again, yours should look a lot better. So now I have my 4 bases all colored. I'll go ahead and assign colors to the deoxyribose down here, and the phosphate group. Okay, so now that that's done, and I'll just be working with this little segment that I've colored, but obviously you're going to be working on your entire sheet. So in front of us, what genetic information do we have? We actually have DNA. And so some of you put something different on your worksheets yesterday, and that's totally fine. I didn't correct you because I wanted you to kind of go through this process. So DNA, this is DNA because it has two strands. So this is one strand and this is two strands. A lot of you said it was RNA because it wasn't twisted, and that was a valid answer. I'm not going to take off for that or anything. But remember that this is basically the DNA molecule but untwisted. Okay? So we have our two strands, and we also, now we know we have deoxyribose, which we know is only in deoxyribonucleic acid. It's specific to DNA. So if we see something that has deoxyribose, automatically we should know it's DNA. So here, I want you to go ahead and label these two strands that we have right here are connected through these hydrogen bonds that we talked about right here. So these right here, zigzagging down, those are the hydrogen bonds. They're very weak, which will come in handy in a minute, but I want you to go ahead and label them however you can, wherever you can. These are the hydrogen bonds. Okay, so they're paired together by the hydrogen bonds. If not, each section or each strand would just be off on its own, doing its own thing, and nothing would work right. We need this double-stranded molecule to happen, to be double-stranded, because of these hydrogen bonds. And what the video just, the last slide ended on was that these molecules are anti-parallel, where anti means against, so they're going against each other. So right here you'll see that we still have the deoxyribose and the phosphate groups, but we don't have the same bases here. So this happens because they are anti-parallel, and it talked about the 5' end and the 3' end. And we're not going to get into like what those are, why they're called that. I just want you to know that always one end is going to be 5', one end on one strand is going to be 3'. So it runs from 5' to 3'. So if this strand over here on the left is 5' to 3', and they are anti-parallel, that means that the right side has to be 3' to 5'. Okay, so they run anti-parallel, and this is a big part of DNA replication that we won't really get into. Maybe in a different biology class you'll get into it, but just know that they run anti-parallel. So say in the future, if I give you one strand and I have that this is 5' and this is 3', you'll be able to fill out the entire other strand starting with that this is 3' to 5'. So going back into our notes where we left off, talking about the DNA structure, this is something that is very important in like a very important discovery in science. And we know our discoveries in science just made by like one person, one time, it just happened and they just happened upon it. No, typically that's not how discoveries are made. Most of the time, discoveries are made through contributions of multiple different people finding little tidbits, and then they piece all those things together. So I did want to talk about a few scientists that are very important to the structure of DNA, the discovery of that. So the first one that we're going to talk about is Chargaff. This man, he did a biochemical analysis of DNA from many different organisms. So he took the DNA from as many organisms as he could get his hands on, and he analyzed them and looked at like the chemistry that makes them up, and he realized that there was very similar things happening in all of the organisms. They were very similar. So looking at that, he found that amounts of adenine equal the amounts of thymine. So they knew what these structures were, and he realized, oh, hey, that's crazy. There are the same amount of adenines and there are the same amount of thymines. And then there are the same amounts of cytosine as there was guanine. So this, he came up with these rules that he found, understood, discovered, and they are the base pairing rules, where G is going to pair with C always, and C can pair with G. It can go both ways. And then A is always going to pair with T, and T is always going to pair with A. You'll never see a G pairing with an A on purpose, and you'll never see a T pairing with a C or any of those combinations. It's always GCAT. And so the way that I remember this is if you just write them down, it spells GCAT. So if you're talking about gangster cats, I don't know, but you can remember GCAT because of the G and the C and the A and the T. So that was a really important discovery. So they realized, hey, there are these 4 bases, adenine, thymine, and then guanine and cytosine are always paired together. So that was a key discovery, and kind of understanding that maybe there's something going on where it's double-stranded. And then Franklin. Franklin, her full name was Rosalind Franklin, so it was a she, and that is very important to her story. So her name was Rosalind Franklin. That is important, I want you to know. And Rosalind Franklin, she, I'll give you a little background on her. She was an expert in her field, and she basically used x-rays to look at extremely small things and like talk about the structure of different things using x-rays. And she was one of like the leading experts in her field. So she worked in this lab, and she got all this work. She discovered all of this stuff about DNA. She actually figured out that the DNA was a spiral shape. So she figured that out, that, hey, it doesn't look like this ladder. It actually kind of twists. So she was the one that figured this out. And then this is a key idea in finding the structure of DNA, which we'll talk about a bit. Because before, they thought maybe it was 3 strands. They couldn't quite figure out how it was working, because back in the 50s, did they have the technology to like see DNA in our cells? No, they didn't. So they were just having to use a lot of chemistry and things like that to figure out like what it was made of or how it was made. So she made this amazing discovery, but you probably have never heard of her name before. And that's because the work that she found, where DNA was a spiral shape, it was actually stolen by the man that worked in her lab. And then that man, his name was Wilkins, and he presented the findings that he passed off to be his own. He didn't say, hey, this amazing lady named Rosalind Franklin discovered this, and I'm just telling you her results. He basically passed them off on his own, as his own. And he presented them at this lecture, and in attendance to that lecture were Watson and Crick. So those names you might be a bit more familiar with. I don't know. There's a chance that you've probably heard of them, because they are the ones given all the credit. So they're given the credit saying that DNA is double-stranded with a base on one side, and the hydrogen bonding the complementary bases of the other side. So they figured out, okay, it's double-stranded based off of Chargaff's findings. And then, after they heard this stolen work in this lecture, they were like, wait, a double-stranded spiral? Like, that could actually happen. And so then they concluded that the shape is a double helix like if you twisted a ladder. And this idea was stolen from Franklin. And so Rosalind Franklin, she was actually given no credit for her discovery that basically revolutionized what we thought about DNA. No one knew about her discovery until almost half a century later. And you'll hear Watson and Crick's name a lot, but they didn't really do much. They just stole their work from other people and gave them no credit. So they were pretty bad scientists, if you ask me. But moving on, so after they figured out the structure of DNA, for a while they had kind of understood the function of DNA, but it took a while before they could actually realize what the function was. They had to figure out how it was shaped first, obviously. So when they figured out the structure, then they could work on the function. So if I were to ask you, like, hey, what does DNA do? You'll probably tell me something like, oh, I don't know, is our genetic information or codes for things, or it makes us us. And all those are right answers, but we're going to get a lot more specific in DNA's actual function. So DNA codes for genetic information, like I just said, in genes, which is the GENES, not like blue jeans. It codes for genetic information in genes, and then it defines genes for you, which is super important to know. Genes are just short segments of DNA. That's important. Coding for specific proteins. And since that's a definition, I'm going to go ahead and highlight it because that's important, and we want to be able to remember that. Okay? And DNA is stored in the nucleus, which hopefully you remember from our cell cycle unit, but it's stored in the nucleus in every cell in your body. Okay, I'm going to just underline that for emphasis. Every cell in your body. So we have trillions and trillions of cells, and in each cell, there is all of the genetic information that cell would ever need. So all of the cells have all of the genetic information, which is a lot of genetic information. But even though it's in every cell in the body, some of the information coded on the DNA. So some of the information like contained in DNA isn't needed in that cell. So all of the genetic information isn't needed in all of the cells, but it's still present in all of the cells. For example, the same proteins aren't used by your stomach cells and your skin cells. So your stomach cells and your skin cells obviously do different functions. They have different proteins that are being produced, and they have different, energy needs and nutrient needs, and all of those things are performing completely different processes. But in the cell, they have the same genetic information. So the way they work through this is that some of the genes in the DNA are turned off. And this is called differentiation. So if you look at the root word here, different, it's basically saying like how we get different cells. So how cells are able to be different because they can turn off and on different genes if they need them or not, which is pretty cool. So the DNA function, it contains the genetic information for the cell, which we kind of already said, but I'm really drilling it into your brain. It contains the genetic information for the cell because it codes for various amino acids which form proteins. So I'm going to draw I'll draw a star around this one because here's another molecule of life. We love them. We live with them. And if you'll remember, on the last page we talked about amino acids being the monomer. Monomer meaning one. So there's one amino acid. But if we have hundreds and hundreds and hundreds and hundreds of amino acids or many of the monomers, then we have a protein, which is the polymer. Okay, that'll be super important for you to remember. And remember, DNA just exists in your cell, but sometimes, like before, when it's preparing for the cell cycle, it will be coiled with the protein, and that's known as chromatin. So just in your cell, it's uncoiled and uncondensed, floating around, doing its thing. But then it coils around a protein known as chromatin, and then it condenses, known as chromosomes, and that's typically during the cell cycle. In humans, this is important to remember, humans have 46 chromosomes, or 23 pairs. So if DNA is needed in every cell in our body, how does it get there? So we talked about it a little in the cell cycle, and we talked about how, well, hey, something has to happen where the DNA copies itself and it goes into the new cell, which is true, and we're going to talk about it a little more right now. So if we're talking about something copying or making more, then we can use this word replication. So DNA replication, we're going to look at the how, where, why, when of it, because it is super important to our cells. So where does DNA replication happen? That is something that hopefully, it makes sense. So where is the DNA located? We already said it's in the nucleus. So, hey, guess what? That's where DNA replication happens, in the nucleus, because that's where the DNA is. So the DNA is not going to go somewhere else to replicate. It has to stay in the nucleus because it's really just too big to leave the nucleus. And when does DNA replication happen? Well, again, we kind of already talked about this. It copies the DNA during interphase of the cell cycle. So remember, the cell cycle is made of interphase, and then mitosis, and then cytokinesis. So an interphase was the longest part of the cell cycle. And it's the longest part because it's just the cell doing its thing. It's going through life. It is starting to replicate its DNA because it knows what's coming eventually. And why is it important to replicate DNA? Well, so that it's in your cells. So the whole point is to produce an exact copy of DNA for the new cell that will be formed by mitosis. Okay, so far we've got DNA being replicated during interphase in the nucleus because we need an exact copy in the new daughter cell. And the how is a little bit more complicated, so let's look at that. So in the how, there are two strands of DNA, obviously, we know that. And those two strands get separated by an enzyme. And enzymes are what molecule of life? Proteins. So it gets broken, the hydrogen bonds get broken between the complementary bases. So the two strands are separated by an enzyme, which is a protein, when it breaks those weak hydrogen bonds. So remember, those hydrogen bonds are super weak. It doesn't take a whole lot of effort for this protein or this enzyme to break them. And then while they're separated, more enzymes attach free bases to the complementary base, which results in two identical sections of double-stranded DNA. So I'm going to draw this on here real quick. I would highly recommend that you do the same. You can do it in one of these spaces over here. You can do it on a separate sheet of paper. But I do recommend that you have this somewhere because, hint, hint, it might come back to you. So let's say this is our DNA. And right here, this is our normal DNA. It's living its life. It's still bound together. So we still got our bonds, or our bases bonded by the weak hydrogen bonds. But something is coming, an enzyme called helicase is going to come and rip those bonds apart so that then they just look like this. And their complement is on the other side. So they're no longer touching. They're no longer bound together. So let's just say, for example's sake, that right here, and you don't have to enlarge this obviously on your paper, you can just write smaller. Let's say we have an A right here. If we're looking at this side, what's going to be paired with that A? Well, it'll be a T. So you'll have a T over here. And let's say you had a T over here. What would be down here? It would be an A. Let's say that you had a G right here. What would be right here? A C. And let's say that you had a C right here. What would be up top? A G. But these are no longer paired together, so then a new strand has to come along, squeeze in there, has to come along and pair the new bases. So like right here, what's going to be paired to this T? An A. What's going to be paired with this C? A G. What's going to be paired with that G? A C. And then if you do it the entire time, all the way down this DNA strand, you'll eventually get two strands that are identical to each other. So this is one strand and this is two strands. So this one will stay in the cell and this one will leave the cell, or vice versa. We really don't know. Okay, so going back up here, usually replication is an exact copy because we want it to be an exact copy, obviously, so that all of the genetic information is the same in all of our cells. So usually replication, or making new DNA, is an exact copy. But occasionally mutations can occur, and these are sometimes caused by mutagens, which we'll talk about in depth a lot more later. And then this bullet right here, where it says strands are complementary, not identical. And that's referring to down here. So right here, we have two DNA molecules with two strands each. So this part right here, we're going to mark it up real fast. This and this are identical to each other. Okay, and we'll do a big problem like this on the board if it's still kind of confusing. These are identical to each other, but this strand and this strand are complementary. And we'll talk a lot about what that means, and we'll get to practice it. Okay, so moving on to the next page. So we know now that DNA is the genetic information in our cells that codes for our genetic information, basically. And the way it does that is through making proteins, which we kind of already talked about, but guess what? We're going to talk about it even more because it's super important. So, basically, protein synthesis goes over like how it goes from DNA to expressing the proteins that enhance your traits and things like that, okay? Because remember, proteins, they have lots and lots and lots of functions, but a lot of them are just to like make up us. So without proteins, we would not be okay. We would not be living. So I like DNA gets all the glory of being like, ooh, our genetics is DNA. But like proteins are really the workhorses. Those are the workhorse molecule. So if we just look at the name protein synthesis, the word synthesis means to make. So protein synthesis is literally talking about making proteins. All we're doing. And protein synthesis, it's called a central dogma in science. So the central dogma. And this is just a process by which the information from genes produces proteins. So this entire idea of how genes happen is summed up in the central dogma. So like I said, we start with DNA. We all know that DNA gets all the glory. So we start with DNA, and then through the process of protein synthesis, we go to mRNA, which we'll talk about more in depth, don't you worry. And then mRNA pairs with tRNA, and because of the tRNA, we get amino acids. And if we get hundreds, thousands of amino acids, then we get a protein. So the summarized version is that we can go from DNA to protein. We have to take a lot of steps in the middle, but we get to go from DNA to protein. And we're going to talk about how. So again, to just reiterate, a gene is a section of DNA. That's super important that we remember that it's DNA. It's a section of DNA that codes for certain proteins. And this is such an important definition. I'm going to highlight it again. And like we already put, some genes can be turned off and on, which leads to differentiation. So the reason that we have skin cells, the reason that we have stomach cells, the reason that we have brain cells is all because they're different and they're able to turn off different portions of their DNA. So like the skin cells aren't concerned with the gastric juices and proteins and enzymes that are breaking down your food. Your skin cells are just trying to do their job, producing melanin and keeping you safe. So, protein synthesis we will talk about, otherwise known as gene expression. I might refer to it as both. You never know. But it's the process by which genetic information is used to produce proteins, because synthesis means to make. So what are we making? Proteins. And protein synthesis, it consists of two processes, transcription and translation. Protein synthesis begins with transcription, and obviously then translation comes. And the way that I remember this, and this is the way that I want you to remember this, you'll be like, well, Miss Sanger, like they both start with a T and then Asian, so how am I supposed to know which comes first? This is how I remember. Transcription has a C right there, so we're going to underline that. And translation has an L right there. Which comes first in the alphabet? Transcription does, so transcription comes first. Okay. So in transcription, the DNA strands are separated. And the complementary strand of mRNA is assembled. Similar to replication, but instead, in RNA, we don't have thymine. It's replaced by uracil. So I'm about to draw an example of this, and you're going to draw it too. Okay, so let's say, I'm going to zoom in a lot. Okay, so let's say we have DNA right here. I'm going to make up an arbitrary DNA strand. It's going to be T, G, C, A, T, C, C, G, A, T, G, G, C, T, A, A. Okay, that's good enough. So that's our DNA strand. In reality, this will be thousands of bases long, but obviously we don't want to do that. We can get the hang of it after, 10 or 20. Okay? So if we have that DNA strand, this is going to be our RNA strand. So anywhere where we put a T, it's now going to be a U. So here, what pairs with T? A. What pairs with G? C. What pairs with C? G. What pairs with A in RNA? AU. A, G, G, C, U, A, C, C, G, A, U, U. Okay, so there's our complementary strand. And when we say complementary, if you're like, oh, that color really complements your complexion. It's not the same as your complexion, but it goes well with it. So A goes well with T and G goes well with C. Except in RNA, A goes well with U. And that's just something that we'll have to practice over and over and over again. And trust me, we will. It's for you to get the hang of it. So basically, this is transcription, and we end up with a type of mRNA, or we end up with a type of RNA known as M or messenger RNA. Or mRNA for short. So, if transcription begins with the DNA strand, I want you to tell me where you think transcription is going to take place. If transcription starts with DNA, transcription takes place in the nucleus. That is super important that we know that. Okay, so this mRNA, this messenger RNA, sends the message from the nucleus into translation. So translation is next. And translation is the amino acids. So remember the monomer for proteins. The amino acids are assembled into proteins based on a sequence of triple bases called codons. And codons are 3 bases of mRNA that code for a particular amino acid. Guess what? I'm going to highlight that because that is so important. It's only in mRNA that we will use the word codons. Codons are only found on mRNA, and those code for 20 different amino acids, okay? So there could be many different pairings of the codons of those 3 bases, but they only make up 20 different amino acids. Okay, so I'm going to model translation for you. Starting with our mRNA from up here, I'm just going to copy that down. I'll keep it the same color. So it's our mRNA, and it's A, C, but now, since we know that they code for in 3s, I'm going to kind of separate them. ACG, UAG, GCU, ACC, GAU, and we have a U left over, so I'm just going to ignore that for now. Okay, so we have our mRNA, and from this mRNA, we are going to be able to get tRNA. And the tRNA is just going to be this complement again. Okay, and so when this happens, it is attached to an amino acid, and this amino acid, when they're all together, will be bound, and these amino acids here that I drew and that I will now label, these are amino acids, and they're bound together. And when you get hundreds of them, you get a protein. And is that our end product? Yes, that's what we were trying to make the whole time. Okay? So this idea of a genetic code, there is a triplet code, what we call, and triple just meaning 3. So there's a triplet code, meaning it's a sequence of 3 nucleotides on a DNA or RNA molecule that codes for a specific amino acid. So on mRNA, we do call it codon, and on tRNA and DNA, we'll talk about what we call it, but basically just know that 3 bases, I'll write this, 3 bases equals 1 amino acid. Okay? And this code is said to be identical in almost all organisms. So we can't say all, but like even organisms, you're like, well, we share nothing in common with a measly little bacteria. No, not true, because the codon for phenylalanine is the same in bacteria and humans. So now that we have a base understanding of DNA structure, function, and replication, and protein synthesis, we will be doing more worksheets and activities with these, but these notes are going to be super helpful for you to complete any assignments or to study for any future tests that we might have.